Hello guys, welcome back to my channel. Today we can look how we can study bacterial growth curve in a very simplified and a fun way. So see, what is bacterial growth curve? Just as the title says, has to do something with the growth of the bacteria. Okay, so how I'm going to plot this bacterial growth curve? I'm going to take some bacteria and put them in a liquid medium, and I'm going to watch the bacteria growing, dying, whatever is happening into the bacteria in a time period, and then I'm going to plot this thing in the graph. That is how you get a bacterial growth curve. So when you do this, the bacterial growth curve will have total of 4 phases. One is the lag phase, then it is the log phase, stationary phase, and the phase of decline. Then you can see your graph is having a red line and the graph is having also a blue line. So blue line denotes your total count. Total count means number of bacteria, all bacteria, whatever is living, whatever is dead, all of them together gives the total count. Whereas the viable count will give only the bacteria that is alive at the point of time. So let's see each phase one by one. First phase is the lag phase. So this is the phase at the point of time from when you inoculate the culture to the time the bacteria become metabolically active. So what is happening during this period? The bacteria is utilizing the nutrients present. He is eating and eating it, and then becoming the biggest of them all. So while he is eating food, he is making enzymes, he is making metabolites. So see, bacteria keep on increasing in size, enzymes and metabolites. See these granules here, it demonstrates or it depicts enzymes and metabolites are accumulating in him. And see, the last one is the biggest and the fattest of all. So maximum size will accumulate at the end of the lag phase. Then we have the log phase. Log phase is the time when the bacteria are dividing exponentially. He is dividing, he is using binary fission or whatever the mechanism he is using to divide, he is using that, and he is dividing very fast. So bacteria is dividing exponentially. Since he is dividing very fast, he is of smaller size. So he is small in size. Now see, I have added some bacteria running around. This is to show that during this phase is the time when the bacteria is biochemically active. So when you are needing to do some kind of biochemical studies, this is the time you should do. You have to wait for the time when your bacteria becomes biochemically active, which is when the log phase. Then see, all of them, I have given all of them a pink color. So they are uniformly stained, and this is also the best time to study gram staining. Okay, best time to perform gram staining. Now we are moving on to the stationary phase. In stationary phase, see, the number of progenies formed is equal to the number of progenies that will die. This is why, because during the exponential phase, they are utilizing all the nutrients, they are exponentially dividing, right? Then comes a point when the nutrients are being lacked. See, our cute little bacteria, he is not having anything to eat, so there is a lack of nutrients. Then see, inside him some toxins, some autolytic enzymes are also accumulating. Toxins are very harmful for his body, and autolytic enzyme, he is also making something that will kill him. So all of this will accumulate, that will lead to the death of the bacteria. And this is also very interesting phase when I have shown you in the last phase that all of them were in the same color, right? So this is the phase when they will become gram positive or gram negative. They will choose to become which I should become and which I should not become. Okay, so this is the time when they will become gram variable. And see this granules that looks like gems, gems chocolate. See, this is the time they will produce various type of storage granules. And also see, there are some kind of things outside this bacteria. So he's putting out its spores. Sporulation is also happening in this phase. So they will also produce some kind of exotoxins, antibiotics, everything happens in this stationary phase. Okay. So lag phase, there will be merely the increase in size, and in the log phase they will divide, multiply, and they will divide, multiply exponentially. And in the stationary phase is the time when they are preparing whatever the bacteria has. Okay, they will become the gram negative, gram positive. They will produce more storage granules, they will produce spores, they'll produce exotoxins, antibiotics. All this happens during the stationary phase. Then after everything, the bacteria has served its purpose. Now he is going to die. Okay, this is the phase of decline. He is dying, so many are dying. Some people are sad because so many of them are dying. So the bacteria here will stop his division and the death will continue. And we can see some involution forms. Involution forms are irregularly formed bacterial bodies. Since there is lack of nutrients, he cannot grow to his preferred size or his maximum potential. So we can see some kind of involution forms also. I hope you have found it very nice to study each of the stages. Now let's just summarize this in the tabular form. Okay, so in lag phase we have seen none of them are dividing, they are just increasing in size. There is no death happening. Total count and variable count are in the flat. Okay, it's flat. So what is happening during this size? There is accumulation of metabolites, enzymes, and they will attain its maximum size. What is happening in the log phase? Here also bacteria is starting to divide. Here bacteria is starting to divide, but no death has ever occurred here. Total count is rising, viable count is also rising. Okay, now they are uniformly stained. I have shown them all of them in pink color, so they are uniformly stained. I have shown them running around, so they are metabolically active. I have also said that they are exponentially dividing, right? So all of them are small in size. Okay, now then comes a stage where there is happening death. That is our stationary phase. So bacteria is still dividing here, bacterial death is occurring here, total count is rising, but the viable count is flat. Okay, this is the time when they are producing their own identity. They are becoming gram variable. Okay, gram variable. They will produce granules, they produce spores, endotoxin, antibiotics, and bacterioricin. Okay, bacteriocin. Okay, then comes a time when the bacteria will stop dividing. So there is no division, death is happening, total count become flat, and our viable count is falling. Okay, then the special feature we have to see here is the involution forms. We can see involution forms. So I hope you guys have found this easier with the pictures I have attached. I hope you can write something in your university paper. So if you found this helpful, please share with your friends. Comment down below which topic you want to study next. Have a nice day.